So we are live streaming, right? Okay. Can everyone see this all right? Okay, again, consent to audio and video recording. This meeting is being recorded and posted onto YouTube. Here's my personal introduction for those who may be joining for the first time. Here's my email, especially if you want to get in contact and ask a question. Feel free to reach out. This is a 15-week full course. We're on week, I believe this is the first day of week 13. 3 lectures per week, Thursday, Friday, Saturday, and off-hour Sunday, all beginning at 8:30 p.m. Eastern Standard Time. We do have a group meet where we have a bunch of the students and all of the tutors. All the lecture recording links and PowerPoints will be posted there after each class. We'll also be posting any updates or changes to scheduling. And it's just an easy way to get a hold of me or any of the other tutors for any questions about any topics or just the MCAT in general. Okay, so for today's learning objectives, we're going to be learning about what carbohydrates are, what carbohydrates are most usable by our bodies, and how do our bodies get energy from carbohydrates. And that's going to be probably the bulk of the lecture. How do our bodies get from energy from carbohydrates, because it's a big topic. In general, this is going to be one of those really long lectures, or longer than normal, because just there's a lot to cover. Because of that, I'm not going to have any poll questions actually today, but I will be stopping to answer questions that you guys have, as well as maybe ask questions for the chat, right? Just because this is a long lecture, I don't want to go like very too long, but there's a lot of information here. And I don't believe that asking poll questions for this topic would be very effective, because this is something that you really have to like think over and like memorize over a couple of days. And having poll questions today is not going to be, I expect probably you'll get a lot of them wrong anyway, just because you haven't memorized the information yet. Firstly, we're going to be just talking about carbohydrates in general and their structure. So overview, carbohydrates all have the same basic framework and are categorized into different groups based on size. So most common example of a carbohydrate right here is glucose, right? All carbohydrates use a ring structure, very similar to this, where they have an oxygen, carbons, and then they have OH groups sticking either down or up. And then it's not shown here, but there's in the opposite direction is pointing an H group, right? So this one's going up, this one's going up, this one's going down. And then at the end here, they have this CH2OH, right? This end. This ring is called a saccharide ring. Shown here is glucose, a monosaccharide. So monosaccharide is only made up of one saccharide ring, as shown here, glucose. This is the simplest form and the form that our body uses the best. However, there are also disaccharides, which are 2 sugar rings, polysaccharides, which are a couple of sugar rings, and then oligosaccharides, which are many sugar rings. Okay, and some of those we get a variety of those from different foods and different carbs, like classic carbs like flour, wheat, things like that. And those give different types of carbs and different types of molecules. And our body generally tries to digest them by breaking them down into this one, glucose, right? Or into something very simple that it can digest easily. So talking about polysaccharides, saccharide rings can be combined together to form more complex sugars, as shown here. A glucose and a fructose can combine to form sucrose, a disaccharide, right? And you see here, this used to be an OH, right? And this used to kind of be an H as well, OH as well. Over here on this fructose, if you notice, this fructose is a little bit different than this one. This is a 5-membered ring instead of a 6-membered ring. We'll get into that in a bit, but they can be 5 or they can be 6-membered, these sugars. And they undergo something called a dehydration reaction, which we talked about earlier when we were talking about peptide bonds, right? If this had an OH, this had an OH, got rid of one from each side, and you ended up with this, right? You end up with one OH and one H gone. So H2O, and then this bond formed like this. All right, and this, I do think you should maybe know this one specifically, this disaccharide. It's called sucrose. And the reason you should know it is because this is table sugar. This is what we usually use to like sweeten our foods, like put in our coffee or make cake with. It's called sucrose. It's a combination of glucose and fructose. So the site of binding is the C1OH of one sugar and either the C4OH or the C6OH of the other sugar. This bond is called a glycosidic bond. And like I mentioned, it's a dehydration reaction to form this bond. In this case, it's a 1,4 glycosidic bond because it's between carbon 1 and carbon 4, right? So you always number starting here and going this way is how you always number. So 1, 2, 3, 4, 5, and this one would be 6. And then again here, start with the O, 1, 2, 3, 4, and 5. Again, 5-membered rings have only 5 carbons, whereas 6-membered rings have 6 carbons, right? And obviously you can't form a 1,6 glycosidic bond with 5-membered carbon because it doesn't have a 6th carbon. So when you're doing glucose with something like fructose, it's a 5-membered ring, has to be a 1,4 glycosidic bond, but there are some cases where you'll make 1,6 glycosidic bonds, and just know that I don't think you really have to know too in depth into the differences between them, like the real details. But no, there are 1,6 and 1,4. Okay. So as I showed before, there's 2 different types of rings. They're called pyranoses and furanoses. Pyranoses are the 6-membered rings, which have 6 carbons, and furanoses are 5-membered rings with 5 carbons. Glucose is the prime example of a pyranose ring. Fructose is the prime example of a furanose ring. Next, we want to talk about how to actually name these. So in the ring format, the determining factor of naming a sugar is the direction that the OH is pointing. So if you see here, some of these OHs are pointing up and some of these are pointing down. This is extremely important because if you reverse it, it's a completely different sugar, completely different molecule, at least in this ring structure. And that's how we name it. So the C1OH is especially important as it determines whether something is alpha or beta. Pointing downwards is alpha while pointing upwards is beta, as you can see here. And you can kind of interconvert these. There are reactions that interconvert these, but this is alpha glucose, alpha D glucose, which I'll cover the D as well in a second, and beta D glucose. For the remaining carbons, the direction of the OH determines the name of the sugar. I should actually say, for these at least, right? For example, for glucose, it's always up, down, down, up, down, right? So you can see here down, up, down, up, down. Wherever you see glucose, it's always going to be down, up, down in that order. If you instead went down, 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 it's no longer glucose, it's a different sugar. For these next ones, 2 to 5, right? And for the final one, the C6, the direction of the C6 carbon group determines whether it's D or L, where up means D sugar and down means L sugar. The majority of sugars you'll see will be in the D configuration because that's the most stable configuration. That's just like the natural configuration. So you'll almost always see like this, where it's D, but just so you know. Okay. And like I said, I don't expect you guys to know this right away, and that's why I'm not asking poll questions. But this is something that you're gonna have to memorize, and it's just one of the things that you'll have to memorize for naming carbohydrates so far. Next, we want to talk about epimers and anomers. I always see at least 1 or 2 questions on the MCAT. It's very similar to this, where they ask you, they give you something like glucose and galactose, and they ask you what are these 2 molecules called, or what is the name of this relationship between these 2 molecules? And it's called an epimer. So two sugars which vary at exactly one carbon are called epimers. Most common example is glucose and galactose, which only differ at the C4 carbon. All right, so like I mentioned with glucose, goes down, up, down, whereas with galactose goes down, up, up. I'm sorry, could you guys mute just? Okay. Thank you. So yeah, so down, 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 up, down, and this one is down, up, up. The only difference is in this one carbon. Okay, and that's what's called an epimer. We'll also be covering these Fischer projections in a second, but you can also see the difference here in this Fischer projection. These are all the same. However, this one red is different. Anomers are epimers, but they vary at the C1 carbon. So like I mentioned, the C1 carbon determines if something's alpha or beta. So if you have an alpha glucose and beta glucose, they are anomers of each other because they are epimers as well, because they only vary at one carbon, but that carbon is at the C1 carbon. So anomer is a more specific, more descriptive word for that. Okay. So next, we want to talk about Fischer projections. Fischer projections are the linear way to draw sugars instead of in a ring. You may be asked to convert from one form to another, and they're drawn like this, where it's the C this way, and then it has this double bond O, this OH. It can be converted into a ring, right, by using this O, forming a ring structure. And you actually may be asked to convert from one way or another. And the way you know which molecule this is based on the OH. Instead of up and down, it's left and right. So you can see here, it goes left, right, right, right. Okay. I could explain like the full process, but I don't think I could do as good of a job as this diagram and this site and this link. I highly recommend, once the PowerPoint's out, to go check out this link, or if you just want to take a picture and copy it, or you can just look it up. Very highly recommend. It makes it very easy. This is how I learned it when I was doing biochemistry for my course. This is how I learned it when I was doing organic chemistry. This is how I learned it again when I was doing MCAT. I'd always come back to this whenever I have sugars. I always come back to this to convert from one way to the other. It's very simple, very easy, and it's laid out in a way that's very nice to see. So I would highly recommend just taking a picture of this and maybe going to this site and reading up on this, because I think they did a good job of just illustrating it and demonstrating it. I'm not going to spend too much time on this, and I don't expect you guys to figure it out right now or kind of memorize this right now, but you guys should have an idea of how to do this, because this is something that they may commonly ask you to do. Okay, so take a picture if you haven't already, or if you want to wait for the PowerPoint to come out. So next I want to talk about, and last I think for naming carbohydrates, I want to talk about the difference between an aldose and a ketose. So an aldose comes from the word aldehyde, and ketose comes from the word ketone. So if you notice, some sugars like glucose have an aldehyde, right? And this is talking about the Fischer projection. You don't see it in the ring. There's another way to look at it in the ring, but the aldehyde is only present in the Fischer projection, because it becomes like this in the ring. So you can see here it's an aldehyde and it's starred for you, and this is the carbon actually they started, which made it very nice in this diagram. And ketose, you can see here, this is a ketone, not an aldehyde anymore. This is called ketose for like something like fructose. An easy way to determine whether it's an aldose or ketose, if they give you a ring, which I don't know if they would, because that's a little bit tricky. They usually, I think, will give it in a Fischer projection, or they'll tell you. But in case they do, if the cyclic form has two CH2H groups, it's a ketose, right? So you can see here and here, or this whole thing, I should say. It's ketose. If it only has one, it's an aldose, like right here. The one we've been looking at, glucose, is an aldose, so it's easy to see. But yeah, just know the difference, especially in the Fischer projection. But I would also suggest learning it here. And I would suggest this is a good diagram just to also learn when you're converting from Fischer to the ring. This is kind of a good idea of what happens essentially. So because they started, so you can see this is this carbon, this is this carbon. You can kind of see the mechanism kind of how it rings up. I didn't show the mechanism here because I just think it's a little bit too complicated and too involved to kind of go over. And I don't think they'll ask you specifically about it, but if you want to, I would suggest looking up the mechanism just for your own like understanding, because they may not ask you, but it may help you to understand this concept better. But this is also a good diagram to show it. Okay, any questions before we move on to our next topic in the chat? Or if anyone wants to raise their hand or anything about anything I covered so far, about just naming about what a carbohydrate is. Okay, if there are none, we'll move on. And if you have any questions that come up, please feel free to share as I'm going through. We have another tutor, Janelle, here, who will answer questions in the chat. So the next topic I want to go over is glycolysis. This is where we're going to begin actual metabolism of carbohydrates, how our body uses carbohydrates to get energy. Glycolysis is the first step, and it's also the step that every living organism uses. It's a basic energy producing pathway that all organisms use, from bacteria, plants, humans, everybody. It's highly inefficient compared to aerobic respiration, which is what we mainly use as humans, but it is faster and it doesn't require oxygen. So sometimes, for example, in our muscles, when you're working your muscles really hard, they'll actually switch to using mainly glycolysis, and there are bacteria and other organisms that don't use oxygen. They don't use aerobic respiration, and they only rely on glycolysis. And there is another more advanced form of glycolysis that they use, but we don't have to go over that because it's not directly tested on the MCAT. MCAT really only tests on glycolysis and the human next steps, or actually the animal next steps. This process involves the breakdown of glucose into pyruvate, and it results in 2 molecules of ATP and 2 molecules of NADH. NADH is a very important molecule in ATP production, which we'll cover next week. Our body directly can convert NADH into ATP through a process called the electron transport chain and ATP synthase. But like I mentioned, we'll cover that next week. And 1 molecule of NADH is equivalent to about 2.5 ATP each. It's a little bit up for debate exactly how much, 2.5, 3, but the more modern literature says 2.5, so that's what I'm going with, and I think that's what the MChem mostly uses, but I think they'll mention it if they ever want you to count. But yeah, 2 molecules of ATP and 2 molecules of NADH per glucose. And like I mentioned, and if you remember from the digestive system lecture we did last week, if you have a complex carbohydrate, our body, before it even gets to this stage, it'll break it down into glucose, into a very simple sugar that it can digest, glucose, and then it'll be given out to the cells to use. So you wouldn't have like something like sucrose going into your cells really, because our body can't really break that down into energy. It's kind of useless. Okay, so in terms of, and this can apply to glycolysis, pyruvate dehydrogenase, and the Krebs cycle, which I'm going to cover today. The steps, I think you should know the general steps and some of the names of the molecules. For biochemistry class, you may have had to memorize the exact mechanism and any enzymes that were used. For the MCAT, you don't really have to know that. It's extremely unlikely that they'll ask you about an enzyme or something like very, very specific about the process, like a specific step, just because it's way too in detail and way too much for you to memorize. But you should know the general steps, what the logic is behind them, what molecule starts and ends, which steps produce ATP or NADH, right? Okay. But you'll see as I go through. So the first step is you phosphorylate glucose to glucose 6-phosphate. If you guys remember, phosphorylating something is kind of like, I want to think of it as activating something. By phosphorylating something, you activate it in most situations, right? So, and also in order to phosphorylate something, you actually need to get that phosphate from somewhere. That somewhere is usually ATP. So you actually take a phosphate from ATP and put it to work somewhere else, right? And this may seem counterproductive. Well, why, if you're trying to make ATP, why are you using ATP to activate? Well, it's because you actually need to use some of it to start the process, but you end up making more than you use. So it's actually a net gain, right? So this first step uses 1 molecule of ATP, but it's necessary to be in the process. Again, this is what glucose looks like in kind of like a linear format. And you just added this PO4, this phosphate, onto this carbon at the end, okay? To activate it to the 6th carbon. Next, we need to move the carbonyl group from C1 to C2. This turns the aldose glucose into the ketose fructose. The current molecule at this stage is fructose 6-phosphate. We'll see exactly like this needs to happen in order to progress, and we'll see in the next step why that is. Okay, just know that immediately you turn glucose into fructose. The next step is making the molecule more symmetric. Since this is no longer an aldose, we can actually phosphorylate the C1 carbon. That's why we needed to change it, because if you remember, aldehydes have a double bond O on the end, which cannot be phosphorylated. So we actually need to make it a ketose and move the double bond O from the carbon 1 to carbon 2, so we can actually phosphorylate carbon 1. So, and this will make it more symmetric, and you can see here, this was originally already here from the first step. We just added another PO group right here onto the C1. You know this is the C1 because this is where the double bond O is, at the C2 carbon, okay? And now you have 1,6-fructose biphosphate. Now that the products are somewhat symmetric, we can now cleave the 6-carbon fructose into 2 3-carbon glyceraldehyde phosphates, right? We can make the one really long 6-carbon chain into 2 smaller 3-carbon chains, each with their own phosphate group. Okay. So you have, technically, there is an intermediate. So when you do this, initially you get one of these, which is the glyceraldehyde phosphate that I mentioned here, the 3-carbon glyceraldehyde phosphate. You also get this, right? Because these are kind of symmetric, but they're not exactly symmetric, right? You can see that these are a little bit different because this has double bond O on the end, this one has in the middle, right? But this will, in the exact next step, this will get converted into this. So you'll actually have 2 of these now. Okay, so initially you get 2 slightly different molecules, but then one of them just gets immediately converted into the other one. So that's why I didn't bother with the intermediate. But I just think you guys should know that. Okay, so you have, in the end, you have 2 of these. Okay. So far, we've had to phosphorylate twice, and we've actually had to use 2 ATPs already, right? So we're at a pretty significant net loss, right? There isn't another phosphorylation event which turns the glyceraldehyde phosphates into biphosphates by adding another PO4 to the opposite side. So again, we're actually phosphorylating one more time for each 3-carbon, right? And since we have 2 3-carbons, we now have to use up basically 2 more phosphorylations. But these don't use ATP. Rather, these use NAD plus as a cofactor, and it actually turns it into NADH, which is what we want. We want to turn NAD plus into NADH. So rather than using energy using ATP, we actually make something NADH, which is equivalent to ATP. So this is a very net positive step. And again, this occurs twice. So 2 NADPH are produced, and NADH are produced. Again, because there's 2 of these, right? And each one gets an extra phosphate. Okay. this phospho group is immediately removed and is used to convert ADP into ATP. So we put the phosphate on by using NAD+, and then we immediately took it off and put it on an ADP to make ATP, okay? This is the actual direct energy making step. NADH, it's technically energy, but it's not an energy making, it's not directly energy, whereas this is directly making an energy. It produces 2 ATP because you do it twice, one for each molecule. Lastly, the first phospho group that we originally put on is removed, and it is again used to turn ADP into ATP. And again, this produces 2 more ATP. Ultimately, this produces 4 ATP and 2 NADH, but we had to use 2 ATP to actually for this process. So we have a net gain of 2 ATP, 2 NADH. Okay. I know there's a lot of like letters, but try and follow, and if not, please let me know in the chat if you want me to explain again. The final product of all of this is called pyruvate, right? And it's shown right here. This thing called pyruvate. Okay. Now, for a lot of organisms, this is somebody has a question.
Mark, is it will they ask us about committed steps, since you mentioned that the MCAT doesn't go in detail?
Sorry, could you repeat that? Which kind of steps? I didn't hear that word.
You said the MCAT doesn't go in detail. Would they be asking us about committed steps and stuff like that?
No, I don't think so.
Okay.
I don't think so. Because that's that's under more, that's very specific, because that also involves knowledge of enzymes, and like maybe you can memorize what are the committed steps, but that's kind of useless if you don't know why they're committed steps, and in order to know why, you would have to know a lot about the specific enzymes, which would be too much for them to ask you, I think.
Okay.
Yeah, so they don't really ask that. Okay, so this produces pyruvate, and for some organisms, this is kind of one of the end stages. It's not actually, but for the purpose of the MCAT, it's one of the end stages of this whole process for glycolysis. But for humans and other animals and a lot of like plants and stuff that will use oxygen, we can actually continue. Once we have this pyruvate, we can actually use this pyruvate and as fuel as well, and break it down even further and get a lot more energy out of it than we can just from converting glucose to pyruvate. Okay. Before we move on, is there any other questions that anyone wants to ask? Okay. And again, feel free to always put them in the chat if you guys have them. Okay. So next, I want to talk about pyruvate dehydrogenase, which is the next step in this energy making process. So far, we've covered anaerobic respiration. This will be the first step that only organisms, or this step that we're going to be covering now is going to be the first step that only organisms that use oxygen will undergo, because other organisms will do something else with pyruvate, which we don't really have to mention. Pyruvate is the product of the glycolysis, and it cannot directly be used in the Krebs cycle. Therefore, it must be made into a molecule that can directly be used in the Krebs cycle, called acetyl-CoA. Okay, and CoA stands for coenzyme A, and it's a really like big, complicated molecule that you don't have to like know what it is, like, or what it looks like, but it's just know that it's really big, and it's called CoA. Pyruvate dehydrogenase is an enzyme that is responsible for this conversion of pyruvate to acetyl-CoA. It's a very complex enzyme with multiple subunits. I'm sure if you took a biochemistry class, you would have learned about it. It has multiple subunits, E1, E2, E3, and they all have very unique functions that you would have learned about, but you don't really have to know that for this exam, for the MCAT. Know that it is only found inside the mitochondria, and so the first step of the pyruvate dehydrogenase and like this process is to transport the pyruvate into the mitochondria, right? You have to get across, you have to go into the matrix of the mitochondria, so you have to get past the membranes. Okay, so this is what the overall formula looks like of the conversion of pyruvate to acetyl-CoA. As you can see, there's a lot of stuff that has to go in. The actual CoA attached to a sulfur group, NAD+, which we're converting to NADH. So this is how we make energy off this step. And then TPP, lipoate, and FAD, which are other cofactors. And ultimately, you get acetyl-CoA, right, which looks like this, which has this formula, and carbon dioxide. The conversion uses 5 cofactors, which are these 3. And however, you only really need to know about these 2, the CoA and the NAD+. Again, NAD+ important because we actually reduce NAD+ into NADH, and that's how we make energy off this step, because it's equivalent to 2.5 ATP each. And CoA is the actual how we get the coenzyme onto the acetyl group, right? And that's why it's called acetyl-CoA. And remember, each glycolysis produces 2 pyruvates. So you actually, whenever you go into dehydrogenase, from 1 glucose, you actually get 2 pyruvate. So we actually, most of the time we talk about per glucose. This whole process we talk about per glucose. So we use 2, we consider this dehydrogenase process twice. Okay. So we're actually producing like 2 NADH. Okay. So we're producing 2 NADH, and that's how we make our energy off this step. And then, yeah, we have to use 2 CoA enzymes and stuff like that. So I want to move on to now the Krebs cycle. Okay. So this is kind of like the general overview of the Krebs cycle. You start off, you can see, because it is a circle, you can technically start anywhere, but where you actually like really start is here with the introduction of acetyl-CoA, which you just made in the last step, enters this cycle right here, and then we go this way, clockwise. Okay, so this is a very, very simplified kind of diagram, and I really like this diagram in particular because it really touches on very basics that you'll need to know, right? So you can see here in these steps, the NADH is coming out, right? This is because you're producing NADH. And you'll see in just throughout the Krebs cycle that the main way you get ATP is not by directly making ATP. So in this one step, you are directly making ATP, but it's just this one step, whereas these 3 steps make NADH. This step makes FADH2, which is also similar, right? And the reason is because, again, they're actually worth more. And 1 NADH is worth 2.5 ATP. 1 FADH, and I'll cover this in a minute, is worth 1.5 ATP, right? These are actually worth more, so it's more efficient to make them. And then feed them into the electron transport chain and ATP synthase, which we'll cover next week. But that's how you actually make ATP, rather than just directly taking a phosphate from one thing and putting it on another thing. You can actually get more out of just doing this instead. And then making the ATP kind of using the synthase. Okay. And here you can see where you guys make CO2, where it comes out, right? And that's how you the CO2 you make, you exhale, obviously. The NADH is, we make energy. FADH I covered as well. And you make GTP or ATP, they're the same thing pretty much, right, in this one step. And honestly, the real Krebs cycle is infinitely more complicated than this. There's the enzymes, they have like a bunch of different stuff going in and out, and like this is not an isolated system. So there's some of these are actually used for other stuff. So like some of these intermediates, like succinyl-CoA, succinate, they're actually used for other things like amino acid synthesis and things like that, but that's like way too complicated, right? And I don't want to like show you guys a super diagram, right? So this is just like the simple basic stuff that you probably should know. Sorry about that. Okay, so just like before, when I go over the steps, I'll just be covering what I actually think is like the important part of each step. I would say each step mostly has like one or two like unique things about it that you probably should know and like you should kind of like memorize a little bit. The likelihood that they'll ask you is not very high, but in case they do, like you'll have an idea of it. And this will get you a good idea of what the Krebs cycle is, like all of the steps of it, without having to memorize all of the enzymes, all of the intermediates, all of like that stuff, right? Like you probably did for biochem. Okay, so firstly, oxaloacetate and acetyl-CoA react together to form citrate. So you need both of these together, and you release H2O and coenzyme A, and you then recycle coenzyme A because it's not efficient to make it every time you want to go through the Krebs cycle by converting pyruvate to acetyl-CoA. So we recycle it, for instance, it's not used up in this. And notice that oxaloacetate is the final product, but it's also the first product, I guess, right? Or the first reactant, because it's a cycle, goes in here, but you actually need both of these, right? Without oxaloacetate, acetyl-CoA can't do anything. And until you put acetyl-CoA from the pyruvate dehydrogenase step in, this oxaloacetate will never react, okay? So you actually need both of these together. And this is also a little more complicated, but there is like enzymes that actually regulate this, right? And specific like binding, like ES bind oxaloacetate first, and has very specific binding that increases binding for acetyl-CoA. And this is one of the ways that you regulate the Krebs cycle, but it's like going into a little bit too much. Just know that they have to be reacted together and they form citrate, which is the next product. The citrate is then converted into isocitrate. Main thing about this step I want to point out is that this step is very non-spontaneous, so it's very unfavorable. So if you put this step in like in a test tube by itself, it would never happen. On its own, it's very non-reactive, and it's very unfavorable for this to happen thermodynamically. But because it's immediately used up in the following steps, which are more spontaneous than this is not spontaneous, it actually like balances out. So if the next step is very delta G negative, this step is a little bit delta G positive, but it'll still go forward just because it wants to go down that kind of like valley of just lowering energy. Okay. So that kind of pushes it forward. And that's why this step happens. That's the only thing really special about this step that you should know. Next, isocitrate is converted to alpha ketoglutarate. This is the first energy producing step, and it produces NADH from NAD+. Okay. So that's the first time we produce energy. And remember again, because you had two pyruvates from the glycolysis, that means you also have two acetyl-CoAs from pyruvate dehydrogenase. That means you actually, for each glucose that you used, you actually run the Krebs cycle twice. So even though one isocitrate goes into one ketoglutarate, that makes one NADH. Because you're running the cycle twice per glucose, you actually produce two NADH from this step per glucose. Okay. Next, alpha ketoglutarate is oxidized to succinyl-CoA. The enzyme used for this is very similar to pyruvate dehydrogenase in form and function. Again, because I didn't really cover the form and function of that one, it's not really that crucial, but you know, they're very similar. Has the same E1, E2, E3 component. They all actually have this fairly similar mechanism. The E1, E2, and E3 components have very similar mechanisms to the pyruvate dehydrogenase. And it uses the same cofactors such as CoA, NAD+, and like those other ones that you didn't have to remember. And it also makes NADH. The general rule, I think, is if it's a at least for the Krebs cycle, if it's a dehydrogenase, that means it's using NAD+ or FAD+ to make NADH or FADH. So if you see an enzyme that has dehydrogenase in its name, at least for the Krebs cycle, know that it's making NADH. Like that's like a general rule. that I would say. If it says ketoglutarate is oxidized to succinyl-CoA, again, something is getting oxidized. That means it's a reducing agent. Just going back to our lecture on that. It's a reducing agent. That means it has to reduce something else. The best thing to reduce is NAD plus into NADH. When you do that, you make energy essentially by reducing that. Okay, so anytime you see something is oxidized, something is reduced, when you dehydrogenase, that usually means it's an NADH producing step or an FADH producing step. Okay, so this is the second step. Again, this makes 1 per cycle, 2 per glucose. So now we're up to 4 NADH per glucose so far. Next, we want to convert succinyl-CoA into succinate. This is an ATP or GTP producing step. This uses substrate level phosphorylation. So this means just takes directly from the substrate, which in this case is succinyl-CoA, just takes the phosphate directly from there and puts it on an ADP or GTP to make ATP or GTP, right? And that's called the substrate level phosphorylation. CoA is again released as well and recycled. Okay, so now we're up to per cycle 2 ATP directly, 4 NADH. Okay, and then the final steps. Succinate is oxidized to fumarate. This process is actually part of the electron transport chain and ATP synthesis, which will be covered next week, like I mentioned, but it's also part of the Krebs cycle. So what that means is actually the enzyme that converts succinate to fumarate is inside the membrane of the mitochondria. Remember, this happens in mitochondria. It's actually embedded in the membrane of mitochondria, and that's why it is both in the electron transport chain, which happens at the membrane of the mitochondria, so it's partially in, partially out, and it's also part of this Krebs cycle. Okay, it involves FAD plus as a cofactor, which is reduced to FADH2, which is equivalent to, again, there's some debate. 1.5 ATP is like the newer value. Some say it's 2, but I think the MCAT uses 1.5, but I think they'll mention it between 1.5 and 2. Okay. And usually this FADH is directly used to send into electron transport chain, like I mentioned, and ATP synthesis. Okay. The next step, step 7, fumarate is hydrolyzed to malate. There's nothing particularly special about this reaction. Obviously, it's important, and obviously there's like, so if you were to do a very detailed dive, there's like an important mechanism behind it, but in terms of like overall, there's not really anything particularly important about it. So that's why I didn't put anything. Last step, and this is the step to go back to the beginning, oxidation of malate into oxaloacetate. This process also reduces NAD plus to NADH. This is the final step. Currently, after this step, we're on 6 NADH, 2 FADH2, and 2 ATP. Okay, just remember that. It also regenerates oxaloacetate, so the new cycle can begin once acetyl-CoA is brought in again. Again, until it's brought in, nothing happens. It's kind of dormant. Then when you bring acetyl-CoA in, the process starts again. This last step is actually very thermodynamically unfavored. So that means the delta G is significantly positive. But like the body does a special thing where it keeps very low concentration of oxaloacetate and manipulates the concentrations in the cell, which drive the reaction forward regardless. Okay. So the final products of everything, right? Adding everything together, we have 3 NADH, 1 ATP, and 1 FADH produced per Krebs cycle. Remember, 2 acetyl-CoA per glucose, so double that. 6, 2, and 2. Add that to 2 NADH and 2 ATP produced in glycolysis, and the 2 NADH produced in pyruvate dehydrogenation. Adding this all up, you get 4 ATP directly, 10 NADH, and 2 FADH produced. With the electron transport chain converting the NADH and FADH into ATP, that is 32 or 36 equivalents of ATP. Again, depends on the source. There's some contradiction, but I think the newer value is 32 rather than 36. So that's what I would suggest probably going with, but 36 is also an accepted value, like a little bit older, but depends what the MCAT wants to do, but I doubt they'll ask you for counting. Okay, they'll usually tell you. So you get 32 or 36 equivalents per glucose used. That's significantly more than if you just got the 2 and the 2 NADH from glycolysis, right? And you didn't use that many new products, right? Because you look at this, you didn't actually really add anything or remove anything. You just put in the acetyl-CoA, which again, you made out of the glucose that you already have, or the pyruvate that you already have. So you have this, and then you add it in. And this, initially you had oxaloacetate, but then you go through this cycle, and then you end up exactly with oxaloacetate. So whatever you started with, you ended with, right? You just use up this acetyl-CoA, which is kind of like the remnant of the pyruvate that you had at the end of glycolysis. You didn't really have to use up that much more ingredients, I guess. And the CoA gets recycled in the end, right? Because the CoA is not in the product. So the CoA actually gets recycled. So you really didn't use up almost anything, right? Does that make sense to everybody? Okay. So that's about everything I wanted to cover. After this, I just wanted to maybe ask a couple of questions in the chat rather than poll questions, just some thought questions. But I know this was like a lot of information, a lot of memorizing that you probably will have to kind of like look over again. I would highly suggest it. If you are interested in learning about the enzymes, I would suggest like doing some research on your own and finding like the enzymes responsible for each step. They may ask you on the MCAT, not super likely, but they may, just in case, if you want to be prepared for anything. Yeah. Otherwise, if you guys have any questions, otherwise I will move on to a couple of questions that I think are very common on the MCAT regarding some of these processes. But I'll leave the floor open to questions in terms of actual material that is for today, but I will be asking questions and answering questions. So if you want to stick around. Okay.
Mark, since it's a cycle, is there like you mentioned, regulation, like so things get backed up? Will they ask us what will happen if they put like a poison at a point or something? Will they get that serious? Because it's anytime I see a cycle, I always think about regulation, like things backing up and whether it will move forward or positively feedback. How would they
that's a great question, and that was actually one of the questions that I wanted to ask for you, and that's something that's very commonly done. Yeah. They'll ask you, say something like, leave this step, either this step or this step. I don't remember off the top of my head. But there's a common poison that actually inhibits one of these. It's not a poison, it's a specific chemical that they use, but inhibits one of these steps. I don't remember exactly off the top of my head which one, but they'll tell you which one it is. That stops one of these steps, and then they'll ask you kind of, if we have a test tube and we inhibit this, what will we have a decreased amount of? And it'll be like citrate. So just to make it like simpler, and like I'll actually ask this question. So if we inhibited this step, right, what would we have? What would be built up in the cell? Would there be a lot of So if they ask something like this, and they kind of asked, what would, out of these, which would not be built up in the cell, right? Something like, it would be something, could be something obvious like malate would obviously not be built up in the cell. Then this is just an example, right? Like they could ask something like this, and like malate would obviously not be built up in the cell because you're not producing malate. This step is inhibited. Whereas something like, but because you are backing up this kind of process, you have a lot of fumarate built up. If you have a lot of fumarate, you'll actually stop the process of succinate going here. You'll have like build up kind of like backlogging essentially of all of these materials, of all these other materials, right? So that's why it would be like kind of like malate, right? It would be there would be less malate, and there would be kind of like a buildup of these other guys. Does that make sense? So that's something that they may commonly ask, and you just have to look at like which steps inhibited, and then you have to maybe think about, okay, because obviously you have the diagram here right now, but you may not have the diagram on the actual test date. So you have to remember, oh, if I'm doing this, like fumarate goes into what comes after fumarate? Is malate after fumarate, or is malate before fumarate? Is citrate before, is citrate after? Is oxaloacetate before or after? Where does everything stand, right? With this diagram, obviously it's easy, right? You can see that just the next step's malate. But on the actual test day, it may not be as easy if you don't have this if you don't really like know the order essentially. And you don't always have to know the exact order, but you should know like a general order. Like for example, you know acetyl-CoA is the first thing that you start off with. You know that citrate is the first thing that's made. You know oxaloacetate is also like the first thing you start off with. Isocitrate obviously comes after citrate because the names are very similar, right? Things like that. Succinyl-CoA and succinate are right after each other because the names are similar, right? So you should kind of have like a general order memorized, I would say. So that's like one very common question that they ask, yeah.
And this is occurring in the matrix or in the inner, where is this going on?
This happens, like I mentioned, it actually happens most in the matrix, but specifically this step, like the enzyme for this is actually in the membrane, right? Because it's also part of the electron transport chain.
In the inner mitochondrial membrane, okay, yeah. I'm gonna just have one more question. Mark, if you could explain what, I didn't get it. You said there's a step from the citrate to the isocitrate, which is very unfavorable, but it's pushed to continue in the cycle because the other reactions are favorable. But how do you explain the other unfavorable step from, I think you said oxaloacetate to acetyl-CoA? How that's also unfavorable. So what is pushing it since, from malate to oxaloacetate?
Yeah. What pushed it is it's because the concentration. So remember, if you remember with equilibrium, if you remember kind of like with equilibrium, you kind of have the equilibrium favors very, very heavily into malate over oxaloacetate, but you will have a little bit of oxaloacetate. And by kind of playing with the low concentrations of oxaloacetate and by reducing like the concentration of that, you can actually kind of push the malate a little bit that way. So it's not favorable, but the reaction gets pushed forward just by the sheer concentration difference between the low concentration of oxaloacetate. If that makes sense. And it has to do with like kind of like the equilibrium kind of like shifting as you like lower the concentration on one side, right?
Great, thank you.
Yeah. So this is, and that's another question actually that they may ask you. For example, they may ask you, this process has a positive delta G. For which reason is the delta G positive? That's another question I actually want to cover. For which reason is this reaction still pushed forward? One of the explanations will be like what I just mentioned with the concentrations. Another will be it's being coupled with another reaction. Another is because it's going to be pushed, like for the explanation for this one, it'll be pushed forward because the following steps are very favorable. And it'll give you those 4 explanations, and you have to determine which one is applicable to this one. That's why I made a point of it. If they give you this step, right? They give you like this step, then it's because the following step is favorable, right? That's the explanation. If they give you this step, it's because of the concentration difference. But also I wanted to mention that for this step at least, what I explained what is actually happening and coupling are different. So one after the other is not the same as coupling. Coupling is when they happen simultaneously, if that makes sense. Does anybody have any questions about that? How coupling and what is actually happening here is not the same.
I thought you said they weren't going to go in details with that, like, ask us those minute details, like the
No, for this, I mean, they might not, but I'm saying they might. This is like quite like question types that I've seen in like my experience. They may not be like super common, but I've seen these, and I just wanted to like. I think it's very easy to like just get these points, right? Like these are like very simple things to like kind of like remember and kind of. If you're learning about the Krebs cycle anyway, this is just like adding a little bit more to it, and it's just it'll get you points, right? So yeah, so like this. Actually, I'm putting here. This is being pushed forward by these reactions after. Coupling is if you, for example, had ATP being converted to ADP. Right? And you did these two reactions at the same time, and the energy produced by this reaction, which is favorable ATP to ADP, actually powers this reaction directly. That's what coupling means. Whereas what actually happens is kind of like the energy goes like this, like that, kind of. Like if this was like an energy diagram, this is kind of what it's like. So that it'll take this small hit, this unfavorable little bit here, just so then it can go super down, like down a lot. Right? That's not coupling, and that's actually what happens. Okay. Does that make sense? So this is being powered by a spontaneous reaction after. Coupling is spontaneous reaction at the same time. Okay. If there are no questions, I'll move on to the next type of thing I wanted to cover. Another type of question that they may ask you is, for example, they may give you a step. Like, for example, let's do, I'm pretty sure I've seen this question before on somewhere. So they'll give you this process, succinyl-CoA to succinate, and they'll ask you what, this is an energy producing step. How does this step produce energy? And it'll be substrate level phosphorylation, oxidative phosphorylation, reduction of NAD plus into NADH, or it'll give some really wrong answer that's not in the Krebs cycle, right? And then I'll ask you which of these is how this step specifically produces energy. And what you would have to know is you have to remember, okay, what are the easiest ways actually, because these all produce NADH, right? These steps, the only ones that don't produce NADH and produce energy in a different form is this step and this step. This step produces through substrate level phosphorylation, so that would be the correct answer. This step produces by reducing FAD into FADH, right? Everything else goes from NAD plus to NADH. And that's another question that you may see, and I'm pretty sure I've seen it before, where it asks you what type of phosphorylation is it? Or what type of energy producing mechanism is it for this step specifically? So that's why I always made a note to say exactly which form of energy is being produced. Okay. Does that make sense to everybody?
I had a general question.
Sure.
Regarding, you mentioned malate if it's not produced, and everything else behind it gets backed up. Does that mean the whole entire process stops altogether?
Yes, yes. So there are actually, I didn't cover this, but there are checkpoints in multiple different parts of this that will measure levels. And like I mentioned, this is not an isolated system. This doesn't just happen on its own. For example, some of these intermediates, I believe this one is used a lot in amino acid synthesis. So this happens, but then there's also this arrow going here into a different cycle completely. And then there's other things that go in and other things that go out. And there's like, this is like a big web, right? And so there's a lot of regulation everywhere, basically, to make sure that there's not too much or too little of anything else. And it's being very regulated very carefully. So if you do have, for example, a stoppage here, things will kind of get backed up and backlogged. And then eventually it'll stop because it'll stop producing. Mainly, one of the main ways of regulation is it'll stop producing acetyl-CoA, and it'll stop the process right here, right? Just by stopping producing acetyl-CoA, or actually directing acetyl-CoA to different things, because again, acetyl-CoA doesn't just go here, it goes like here, goes here, there's a bunch of different places. So it just directs acetyl-CoA to different spots.
Okay, got it. Thank you.
Yeah. Okay. Any other questions? Okay. Another thing, let me see if I
Yeah, can you leave that so I could just, let me get that link once. Could you go back to that link so I could
yeah.
grab it real quick?
This link, if we'll be posting the PowerPoint in the GroupMe, so if you want to check out the GroupMe, it'll like have the link and you can just click on it. If you want to go straight there, if you want to copy it, sure. Go ahead. But
those projections, Mark. What's the Hawthorne? Is that the boat and chair? Would they give us boat and chair, like recognize glucose?
With chairs, chairs are not common, are pretty uncommon, but they may, I would say. I think that's like a maybe. I think that's a maybe with chairs.
To kind of know all the linear, the boat, the Fischer, and the sick, know all the forms basically.
If you know what a chair is, then if you know what a chair is, it's not very difficult to go from this to a chair, because chairs are essentially like just the very principle behind like chairs, and the only reason they may test it is just to test your knowledge of whether you can convert from a hexagon to a chair, or like this hexagonal form to a chair form, and which you should be able to do if you know what chairs. If you don't know what chairs, how to convert from a hexagon to a chair, then you're not going to know this. Basically, if you know how to convert from any other hexagonal molecule to a chair, or vice versa, you can go from glucose to a chair as well.
All right.
Yeah, so this being like it being a sugar does not actually add any difficulty to the question if they ask that, compared to like any other hexagon or like ring. But yeah, this is like a really good way to just remember it. It's very helpful. Okay. So yeah, just this is a common thing, just going between the rings, and like the rings and Fischer projections, like fairly common. This is a question I see a lot. So they'll give you like glucose and galactose, like I mentioned, and they'll ask you what is the relationship between these two, and they'll say they are anomers, they're epimers, they're whatever, they're isomers, they're something like that, right? And if you don't know the definitions of all those mer words, the words like that kind of end in mer, you'll get caught up. And I know that was like something that I had a little bit of trouble with in the beginning until I like memorized all of them. So yeah, I would suggest learning all those like mer words: isomer, epimer, anomer, all those. Okay.
Mark, there are times I see beta D14. What is the extra? I know you said beta means it's up, the C6 is up. What's the D? Like they'll say beta D, or what's the D for? Oh, okay, there it is. Alpha
Yeah.
D.
Oh, actually, this one doesn't. I don't think this one has D. It's unfortunate.
Yeah, you said it's right.
It has. Yeah, it has MR.
See that D right?
I think the site. I don't think this picture has D, but I think the site has D. So, like, if you go to this link, but I'll just show it here. Yeah, right here, the direction of the D6 DRL.
The direction of the C6.
Of the C6 carbon of
determines
this.
whether it's up or down. I thought that just determines the alpha or beta. So what's the D? What is the D determining?
So D is this one. Alpha beta is this one.
Oh.
This one's, yeah.
Oh my gosh, I did not know that. Okay.
Alpha beta. Add these. Yeah. But like I mentioned, you'll almost always, I think, see D. I don't think I've ever seen a non-D sugar. But just in case, just to be safe. Okay. Any other questions from anyone? Aldose ketose actually also like somewhat mentioned. So that's something like you probably should know, like both mainly in this form, in the Fischer protection where it's easier to see, but just in case, I would suggest this form as well, the ring form. Okay.